Hello, hello. Janae Carruthers here from Science Speak Spokane and also Spokane Falls Community College. I'm here today to teach you another laboratory procedure. This is a standard procedure in a lot of labs. We're talking about gel electrophoresis today, and we'll get to in a minute what exactly gel electrophoresis is, but I want to engage you. So I'm going to give you a few minutes in the chat. Type what you already know about DNA. So in the Zoom chat box, what comes to mind? What do you already know about DNA? What are some vocabulary words? What are some concepts or ideas when you think about DNA using the Zoom chat box? Go ahead and take some time now. Share with us. What vocab words? What ideas? Put in the Zoom chat. What are some vocab words? What have you already learned, or what do you think of about DNA? Go ahead and wrap up your ideas, put them in the Zoom chat so we can all see them. All right, thank you for your inputs. So we may have already learned a little bit about DNA, but let's go over and let's talk about some structure. So when we're looking at DNA, the image up on the left may be that typical double helix that you think of when you think of DNA. The reason it's called a double helix is because it has two strands, and then they're made up of what we call these bases. So humans are made up of bases called C, A, G, and T. We have four bases that bind together, calling base pairs. So some of you in the chat, for what you knew about DNA, you may have put the word cell, and you are absolutely correct. Here are some human cells. Within the cells, especially when cells are replicating, they form the structure called a chromosome. The chromosome separates out into genes. You can see the DNA structure here again. None of this can be seen with the naked human eye. This is our double helix DNA strands. And here are our base pairs, again, made up of C, G, T, and A. Those stand for the four different base pairs or component molecules that make up humans. Now, to date, there is no technology that can sequence the entire human genome. So that means there is no technology as of right now by itself, just like in one run, that can tell us the human is made up of T, C, G, A. You know, it doesn't know the alphabet that may be a human. But with the technology of PCR and the technology of gel electrophoresis, they have sequenced the human genome and DNA into segments and then piece that together to give a good idea of what the human genome is. So the human genome it's about 100 million to 300 million base pairs. This blue image here is showing, about 8 base pairs. So in order to see what a human may have, we use gel electrophoresis to break up segments or fragments of the DNA, and then we can piece that together to make up the entire human genome. It was right around 2007 when the human genome from its entirety was known. And again, that's about, I'm just going to say 200 million base pairs. Humans are roughly 99% similar to each other, but there is variation between each human. And so it's interesting to know this for genetics. It's interesting to know this, for what you're, how parents produce offspring and also disease susceptibility. So this is sort of the concept of using lab protocols in order to answer questions we have, to give answers to observations we have. And these are really exciting ways that we can look at this. So this is gel electrophoresis. This is gel electrophoresis happening in a lab. The image on the top, there is a physical gel, just like you would think of for Jell-O. So this grayish square is a physical gel. We'll show you in a video what this gel means. This is just a gel, and there's a liquid that runs through it. So it sits in a plastic tub, and a salt solution is run through it. There's an electric current, and then there are these wells or little dips or troughs that a scientist could use a pipette to insert these DNA samples in. So you see this liquid looks sort of purple. We can't see with our naked eye, but there are hundreds and hundreds of DNA samples in each of these little wells. So the purple liquid is a DNA sample, and you're putting a DNA sample in each of these wells. The image on the right is the result after a gel electrophoresis is run. So it uses fluorescent UV light, and all those bars, the glowing bars, are different lengths or different fragments of genomic data. So this is the results of what a scientist would see with gel electrophoresis. Here's a simplified version of that UV gel, that result gel. So simplified version, this black box is the gel itself. These squares at the top are the wells or the holes that you would put your DNA samples in. Typically on the edges, scientists will put what's called a ladder. That is the standard. So it is well known amongst the community that if you use a gel of a certain recipe, so a certain gel of consistency, certain recipe of a gel, there's a standard ladder that you can purchase, that if you see a line at this length, that is 3,000 base pairs. Remember, this BP is those base pairs, those A's, those Z's, those G's, those T's in your DNA. If you see a line at this length, that is 1,500 base pairs. And if you see a line at this length, that is 500 base pairs. So again, this is the standard ladder. That's why it's not numbered. It's usually on the side. These are labeled lane 1, lane 2, lane 3, and lane 4. So take a look. These thicker white lines are the samples themselves. These are the DNA samples and how they came out. So the lanes, your answer is going to be 1, 2, 3, or 4. Which lane has the largest DNA strand? Which lane has the largest DNA strand? So the largest DNA strand will be in lane 1. It's going to be more than 3,000 base pairs. So it's above, this is over 3,000 base pairs. Which lane has the smallest DNA strand? Which lane, 1, 2, 3, or 4, has the smallest DNA strand? It's lane 2. The DNA strand in lane 2, so this is the result, this is the DNA, is even smaller than 500 base pairs. I'm not sure of the exact measurement at this ladder, but it is less than 500 base pairs, really, really small. Lane 3 has 2 results because there are strands of 2 different lengths. There's a strand of roughly 1,500 base pairs, and then there's another strand that's a little bit bigger than 1,500 base pairs. So this is a simplified version or result of what a gel electrophoresis looks like. Now, Science Speak Spokane, we promote STEAM at home. So all of us may not have access to sequencing DNA. We may not have access to DNA samples at this number, but we do have access to food dye. So food dye, just your generic kitchen food dye, can be found in any grocery store. We have the chemical components. So this was found through a resource online. These are the chemical structures of dye red 3, the chemical structure of red 40, chemical structure of blue 1, chemical structure of yellow 5. Again, these are just some industry dyes. You'll see that there is a molecular weight, MW, molecular weight for red 3 is 879.86, and it has a charge of -2, so it does have a polarized charge. Red 40 has a molecular weight of 496.416 with a charge of -2. We'll talk about those in just a minute. Remembering that electrophoresis is using a gel matrix to separate molecules by size and charge. Remember that liquid that I showed you that the gel sits in? There is an electric current that runs through the gel itself. So an electric current runs through the gel. So thanks to a resource that I found online, they looked at McCormick food dye. And this scientist found that the red McCormick food dye is composed of red dye 3 and red dye 40. I went ahead and did the math for you. So together, the red dye has a weight of 1,376.3. The yellow has a weight of 1,030.8, and this is rounded to the nearest tenth. The blue dye has a weight of 792.8. Green dye has a weight of 1,327.2, and I just did the math for these 4. So if we were to run these food dyes based on the weights given here, and if we were to run it through an electric current, question, which direction will they migrate? So we have a charge. Think of a battery. The battery has a positive charge and a negative charge. Which direction will these dyes go towards? So if they, they sat in the middle and you ran a current through it, would they go towards the positive end or the negative end? These dyes, we are knowing that they have a charge of a negative charge. Opposites attract in electricity, opposites attract. So the negative charges, they would run towards the positive charge. So we're not multiplying them, we're adding them. So we're making a negative plus a negative still equals a negative. Which color dye will move faster? So which color, if we look at red, yellow, blue, or green, which one will move fastest? Here's their weights. I just put the math for you. Which color will move faster through the gel? Looking at the molecular weights, the blue dye will move the fastest because it has the least amount of weight. The smallest size, the shortest fragment will move the fastest. So I went ahead and you get to see my home kitchen. Next, we will watch my gel electrophoresis at home video. Please excuse the goofy technology, and we'll take a look.
All right, I am ready to make our gel for our gel electrophoresis. In our kit, I have some what's called agar. Agar is a seaweed extract. You can think of it as like Jello. It is a component that you want even distribution of particles. It is semi-pliable, so semi-movable, and it is a component that you heat it up to make it in liquid format. So sort of like Jello, you heat up Jello to make it in liquid format, and then you let it solidify. So you're gonna let it solidify, and then we're gonna run our particles through this gel. And what happens is it's sort of like crawling through a ball pit. Again, if we can make a ball pit as evenly distributed as possible, the smaller particles, the smaller children, will be able to move through the ball pit quicker. So smaller particles will be able to move through the auger particles quicker, whereas larger particles, larger children, are not able to move through as quick. So it separates out the size once we solidify our gel and run it through our electrophoresis. So I have my protocol here. That's what I'm looking at. And it says add 100 milliliters of water to a microwave safe container. So I just happen to have a measuring cup at home. You could use any microwave safe container and measure out, you can measure out cups or you can measure out milliliters. So I'm going to get 100 milliliters of water. And I just have that in my measuring cup. I'm looking at my measuring cup. There is a particle in here. When you're looking at measuring liquid, you always want to hold it to eye height. And the meniscus, or the water level, should be right at the level of the measurement you want. So you always want to hold it to eye height. I do see there's some particles in here, so I'm going to dump and re-measure. I want to make sure it looks like water. This is what happens when you're in a kitchen. I think I made rice the other day with this. So now my hand isn't necessarily steady, so I'm going to set it down and go to eye height and make sure that is at 100 milliliters. I need a little bit more water. Add 2 grams of agar. So I need another measuring dish. 0. So 2 grams of agar. So again, here's my agar. You can find it at sometimes ethnic grocery stores. You can find it online. And I'm going to measure out 2 grams. 2 grams. Haha, 2 grams on the dot. Okay, put my agar back. So 2 grams agar. Adding my official labeled Tang bag. Thank you, Kat, for labeling my Tang. 0.8 grams. Okay, now that I have my measurements, I'm going to go over to my microwave. And my procedure says microwave at 1-minute intervals, so making sure it mixes well. Let me move my camera so you are able to see my kitchen and my microwave and my dog kennel.
So I am microwaving at 30-second intervals to prevent it from boiling over. And I'm being careful with the heat, and I am swirling to ensure all the solid particles are mixing in with the liquid. And do not mind my very dirty microwave. This is real world happening. So every 30 seconds I check, and I'm making sure, ensuring the particles have mixed in, so it's a consistent mixture throughout the entire gel. And now, once it cooled enough to touch, I am pouring the gel into a Tupperware lined with clear wrap or saran wrap. And And the saran wrap is simply so it doesn't stick to the Tupperware itself. So just taking the gel, and it's going to dry. So at room temperature for about 30 minutes, it will dry and solidify just like Jell-O does. Okay. So now I'm making the buffer or the liquid solution the gel will run through. We'll put electric current through. So it is the same material as the gel itself without the agar. So it is Tang, which has sugar and salt molecules, and water. The sugar and salt molecules will hold the electric current running through. And I'm going to mix this, we call it a buffer solution or this liquid. I'm going to mix it without heat, so ensuring it is very well mixed. So we call this a buffer solution. Again, just a solution where there's particles to charge. Here we have our food dye. And we have corn syrup. The corn syrup helps add viscosity to the food dye because the food dye itself is very liquidy. That's an official scientific term. It's very liquid. So we're adding corn so we can add some viscosity and there'll be some weight to the food dye itself. So I am adding a separate food dye color to a separate container. And this is all items you can find at any grocery store. So adding the food dye, again, 1 container per color. So I'm looking at what are the chemical composition, what are the sizes of these 4 different colors. So I started off with blue. You can see mixing the blue food dye with the corn syrup. And there's red. Food dye. Okay, so I'm taking the gel, it has solidified now, and I'm peeling it, peel it off the clear wrap itself. So you'll see it's just like really hard Jell-O, just a gel. And I'm going to put this gel in a Tupperware that's about 3 times its size or its length. This is a new Tupperware. And I am taking aluminum foil and the metal components of this aluminum foil is going to be able to hold that charge. So remember, aluminum is a catalyst. And so this is what's going to add the charge directly into my gel box. This is a gel box being made at home. So adding that aluminum foil, using the metal components, and putting on each side of my gel box. Okay, so our next step, we need to make our wells. So for our at-home kit, I have a fettuccine noodle. That is the thickest noodle. You could also use a blunt skewer. And I'm going to take this fettuccine noodle to my solidified gel, and I'm physically making a little punch or a little, they're called wells, a little indent on the gel. Now in the science lab, there is a standard, it's called a standard comb that makes these wells. And here I am just using a fettuccine noodle, and I'm going to make 4 wells into the gel. This is where we would place the dye, or if we had DNA, we would place the DNA. And the DNA in a scientific lab is typically colored with some dye, and so we can order to see the item that we're putting in. So you can see now we're going to add the buffer. Remember, this is the buffer that has tang and water. So it's water with some electrolytes, water with salt and sugar to hold a charge. And we have some 9-volt batteries. Remember, there's a positive and negative end on the 9-volt batteries. And you could use any number of 9-volt batteries, but in order to have the most power to make an external power source, I am going to plug together 10 9-volt batteries, negative into positive, and make a power source. So now I have a positive end exposed and I have a negative end exposed. So here I'm going to use the fettuccine noodle again. You could use a pipette, but I'm using the noodles for at home. And I am going to take this blue food coloring, and I am inserting this blue food coloring. I'm being very certain. I'm looking, finding the first well, and I'm going to insert the blue dye into our first well. You want to make sure that the blue dye sticks inside the gel. You will see some of the dye diffusing out into the liquid, a little bit of it being mixed around, but you want most of the blue dye to be inserted into the gel. I am using a clean end of the fettuccine noodle. This is the red food dye, and same idea. I'm going to move on to the next well. And I'm inserting the red food dye using the noodle into the well. And again, a little bit diffuses out, but I want to ensure that most of the dye, into the gel. And adding each color into a new well. This could be also DNA samples or particle samples. You have 1 sample per well. Again, using a new tip, pipette tip, or official noodle, and inserting this into the gel. And again, just making sure the food coloring dye is soaking into the well, the gel particle. Again, some is diffusing, but a little bit of diffusion is normal. And then the last color I used is orange. And I'm just looking over, making sure I can see the well. My concentration, with my tongue out. And I'm adding a little bit more orange dye because I wasn't sure if I added enough. Inserting more orange dye.
So DNA is negatively charged, so it runs negative, positive.
So as you can see, I'm talking to myself, but I'm showing the negative and positive ends of my power pack. And again, DNA is negatively charged. So you want to put the positive end. I'm using alligator clips and a wire. Putting the positive end on the aluminum foil, and I'm laughing at myself because I'm mixing up my positives and my negatives. DNA is negatively charged, so it's going to run towards the positive end of the gel box. The only thing that would happen if we flipped our positive and negatives is the DNA or the colors would just run on the short end of the gel rather than the long end of the gel. So here we have our 4 wells with the 4 colors sitting in. Yes, they diffused a little bit, but you can see mostly the color is sitting, and this is how the gel is going to sit. So here we have our gel electrophoresis at-home kit. You can see our 4 wells. So we have our blue, our red, our green, and our orange. The orange is hard to see, partially because it could have been the amount of orange dye I put in. It could also be because the dye and the buffer itself are orange. So I went ahead and took some pictures at different increments. So here is 1 minute. And remember, we had the negative end of our power pack made out of batteries, alligator clipped with a wire to one end of the aluminum foil. So here was the negative end, and on the other end, where the long Y's of the gel is the positive end. And we can see our 1, 2, 3, 4, our wells and our colors. Yes, some is diffusing, but you can see also where the wells are and where majority of the dye sits. So this is 1 minute of holding that electric charge and letting the batteries run power through the buffer or the liquid. Here's 5 minutes. Here's 15 minutes. Now 20 minutes. Do you see the little squares of dye moving? So here's that well, the well, each well. And then here's orange. Orange is the hardest to see, but you can see the dye itself moving in a little cluster. So here's 30 minutes. Here's 45 minutes. Now these little bubbles now at the end are just human imperfections in the gel. It happens, human imperfections in the gel. Here you can see the 4 wells on the left. Bubbles on the tinfoil ensure that, yes, there's an electric current running through. It's not dangerous. The liquid could get warm after running electric current through it for lots of time, but at this point, the liquid was not warm. And you can see the different colors. So the orange is hard to see, but there's orange. We have green, red, and blue. So if I'm looking at these 4 dyes, which are the smallest chemical components and which are the largest chemical components? So in this case, in this dye pack, and again, this dye pack, I believe, was the festive dye pack, whatever that means. In this dye pack, the orange and the red are the smallest because they traveled the furthest. Green and blue are the largest, so they traveled the least distance. So this is our gel electrophoresis at home using food dye. If we were to go back to our DNA particles, this is an example of gel electrophoresis in the lab. On either side, so on the left side and the right side, are again those standard ladders that you can buy. The standard ladders that are a set length, known length. So here we have 3 kilobases, kilobase pairs. A big K means 1,000 or kilo. So 3 kilobases. If we go further down the gel, we have 1,000 base pairs, or 1 kilobase, 1,000 base pairs. We have 500 base pairs and 100 base pairs. So remember, the further down a particle or a DNA strand goes down the gel, the smaller it is. And here are some known in the middle, the 5 really bright lights. The scientist has labeled the DNA fragment lengths for us. Now, again, why would we want to use these? We could compare species and such as bacteria, different species. We could compare genome or DNA length. Also, gel electrophoresis is used again to answer questions, observe different phenomenon in DNA or chemical size. It can also be used in forensics. This picture is an example of DNA itself, but what you could have is in one lane, the DNA found at a crime scene It would not, and then you could put suspects in following lanes and compare, a pretty inexpensive way to compare DNA and DNA found at crime scenes. It could also be in forensics. So here I hoped you learned a little bit more about base pairs. I'll go ahead and go back to our DNA structures. So back to our base pairs, which remember are these chemicals, the G's, the C's, the A's, and the T's. And the human genome is about 200 million base pairs. So these DNA gel electrophoresis are just a small example. What happens is scientists chop up the DNA, code it a bit at a time, but we can also use it to examine for forensics. We can use it to examine bacteria and viruses, and also in diseases. So this is what we learned today. Thank you for your time.